Good morning. In this video, we are going to pursue our discussion and explanation of concepts that are related to the water potential. And here we discuss tonicity. And tonicity is rather very close to osmotic, solute potential and to osmotic potential. So tonicity is very close to solute potential. Let's see the definition of tonicity. Tonicity is the ability of a solution to make water move into it or out of it by osmosis. So it's very close to solute potential, because the more you increase the solute potential, the more water that flows inside the solution. Here we are specifying an extracellular solution, but this applies to any other solution. And tonicity is close to solute potential, but it is different from osmolarity. Up till now, we haven't yet discussed what osmolarity is, but osmolarity is the total concentration of solutes in a particular solution. For example, 100 grams per liter. Osmolarity per se cannot help you to understand in which direction water will flow. Why? Because it does not take into consideration the difference between two solutions. Because in order for water to flow, there should be a hypotonic solution and a hypertonic solution. There would be a solution that has more water concentration to a solution that has less water concentration. So solute potential and tonicity help you decide, because the solution that has less water concentration is a solution that has a higher solute potential and that has a higher tonicity. Then when we talk about osmolarity, no, it doesn't help us determine the direction of water flow, because this osmolarity just describes one solution and not the relative concentrations of solutes in the two solutions that are separated by a semi-permeable membrane, and therefore does not determine the relative concentrations of solutes on both sides. The presence of solute particles on one side having a particular tonicity will determine what? First of all, it will determine the direction of diffusion. We already know that water will flow from the hypotonic solution to hypertonic solution. So there is a difference or a gradient in tonicity between them. The rate of diffusion, because the higher this difference or this gradient, the higher the rate of diffusion. Tonicity actually is a measure of the difference in solute potentials, or it is a measure of the difference in water potentials and the osmotic pressure gradient. So although tonicity per se is not water potential and is not the difference in water potential, yet tonicity is a measure of the gradient that is present between the two solutions, and therefore the difference in water potentials between the two solutions. Because when you talk about tonicity of two solutions, so tonicity, say we are giving numerical values, so you're going to say that tonicity of water solution is 10 and tonicity of water solution is 30. So this just indicates you that tonicity of solution B is higher, that solution B contains more solute particles, that there is a difference in osmotic pressure between the two and in solute potentials, and that there is a difference in water potential. So tonicity is an indication, rather, of the water potential. And therefore, when you consider the tonicity of two solutions, I mean that 1 and 2 apply only when you are considering two solutions at the same time. The difference in tonicity is actually a difference in water potential and is actually an indication of the osmotic pressure gradient, which will determine the direction of flow of water. So there is nothing new here except for the nomenclature. Here we have three solutions. When we say hypotonic or hypertonic or isotonic, we usually compare between two solutions. So if I say that this solution is isotonic, it should be isotonic to another solution. So it should be isotonic to solution A, for example, or B or C. Hypertonic means hypertonic to another solution. And in such a case, it would have more solute than the other solution. Hypotonic, it means hypotonic to another solution. And therefore, the hypotonic solution will have less solute. Of course, if we want to say it in terms of water, the solution that has more solute will have less water, and the solution that have less solute will have more water. Now, how can we apply this to red blood corpuscles? Now, red blood corpuscles are present in plasma, and red blood corpuscle, we consider the cells in general, including the red blood corpuscles, that they should be present in an isotonic solution. Actually, the verb is here is not the most appropriate verb. We had better say the cell should be present or should lie in an isotonic solution. Because when it lies in an isotonic solution, and this is important to know, when we have a hypotonic solution that is separated by a semi-permeable membrane from a hypertonic solution, it's true that the overall flow of water is from the hypotonic side to the hypertonic side. But this does not mean that at any time there is a small amount of flow in the opposite direction. And when the cell, like red blood corpuscles, they are not nucleated, but the same applies to red blood corpuscles. When they are present in blood, they are surrounded by the watery environment of the plasma. So all the time, there is water that flows in and water that flows out by osmosis. But at any time, the rate of inward flow is equal to the rate of outward flow. So the content of water in the RBCs does not change. But if the cell, on the other hand, is put in an isotonic solution, what would happen is that the hypotonic solution has, by definition, more water than the inside of the cell. So water will pass, and this is how we should explain it. We should explain it explicitly. So water will pass by osmosis from the hypotonic surrounding of the red blood corpuscle to the inside, which results in increased volume of the cell, and it might result in bursting. And if red blood corpuscles burst, we would have anemia, or a diminished number of red blood corpuscles in the body, which just implies that there would be less oxygen carriage to the tissues. And that's exactly what happens when you infuse fluids to the patient, large volumes of fluid that are not isotonic with the blood. For example, you cannot give an intravenous infusion of pure water to a patient. He would die on the spot because you would make the plasma more hypotonic, resulting in bursting of the red blood cells. Similarly, you cannot give the patient a hypertonic solution because in such a case, the plasma would be, of course, I mean intravenously, the plasma would become hypertonic, meaning that it would have less water than the inside of the red blood corpuscles, which would result in the outwards flow of water from the inside of the red blood corpuscles to the outside. The red blood corpuscles would become crenated or shrunken, and this diminishes the function of the red blood corpuscles, being unable to carry oxygen to the tissues. The red blood corpuscles should stay in an isotonic solution. What makes the cells burst is that these are animal cells. After all, they are not plant cells. But what happens in the plant cells is something different, because If we consider the plant cell, it has a cell membrane, and that cell membrane is surrounded by a cell wall. All this thickness around the plant is the cellulose cell wall. And then inside, we have a large central vacuole that is full of solid particles and water. If the plant cell is put in an isotonic solution, there is no problem. If it is put in a hypertonic solution, the hypertonic solution will withdraw water from the sap vacuole, and the plant cell will wilt. It will be shrunken and it will wilt. But if the plant cell is put in a hypotonic solution, it will absorb water as usual, like in the animal cell. But the differences is that it will not burst, so there will be no bursting. There will be just swelling and increase in size of the sap vacuole. When the sap vacuole increases in size, there is no bursting because we have a protective cell wall. But what happens is that this swollen sap vacuole will have increased pressure on the wall, on the cell wall. So there will be increased hydrostatic pressure inside the sap vacuole. This will make the plant cell turgid and stay vertical and upright. And this means that we have increased turgidity, and therefore the hydrostatic pressure is called in such a case the Turgor pressure. So the Turgor pressure is the hydrostatic pressure that provides the plant with turgidity. So the Turgor pressure is the hydrostatic pressure. It is, of course, not the normal, regular hydrostatic pressure, but it is increased hydrostatic pressure resulting in a fully swollen plant or a turgid plant. So no rupture of the cell membrane occurs because the cell wall will support the cell membrane of the plant cell. Let's read the slide in order to see whether we have missed something. So in example 2, the cell is in a hypotonic solution. The concentration of solute in the beaker is less than the concentration of the cell, meaning that the concentration of water is more. So water flows into the cell, resulting in swelling and bursting. If it is an animal cell, it will burst, but if it is a plant cell, the cell wall will resist bursting, and the cell will merely swell or become turgid. This turgor pressure, or the hydrostatic pressure leading to turgidity, is what keeps the plant like celery crisp. If a plant loses too much water and is dehydrated, of course, the turgid pressure will diminish. When the cell is put in a hypertonic solution, we mean, of course, an animal cell. The concentration of solute in the beaker is greater than the concentration of solute in the cell. Water will flow out of the cell because water flows, as you know, from a higher water concentration to a lower water concentration. And now we are going to discuss how changes in turgor pressure of the plant can occur. Now let's remind ourselves what turgor pressure is. Turgor pressure is the hydrostatic pressure exerted by the fluid of the plant cell, pressing the cell membrane against the cell wall and resulting in turgidity of the plant. So if we read the definition carefully, so we start by pressure, but actually we should say it is hydrostatic pressure because it is pressure exerted by a fluid. So pressure exerted by a fluid is called hydrostatic pressure. And when we talk about turgor pressure, that turgor pressure should be in a plant. And that's why we say that the turgor pressure presses the cell membrane against the cell wall. Why does it press the cell membrane against the cell wall? Because the direction of the hydrostatic pressure is always outwards, and that pressure is what makes the living plant tissue turgid or rigid. If there is loss of turgidity, this results definitely from the loss of water from the plant cells, and this is what we call, or this results in wilting of the flowers and the leaves and different parts of the plant cells. We are not going to discuss this diagram, the one below, because it's talking about aquaporins or water channels, and we're going to discuss aquaporins later on. What I can say is that water passes from one cell to another until equalization of the water concentration on both sides, but there are other substances that pass partly by passive transport and partly by active transport, like sugars, like ions, like amino acids. And there is a structure that is, let's see the key of the diagram here, because we might forget to get back to it. We have the plasmodesmata. These arrows represent the plasmodesmata. And we all know that the plasmodesmata are protoplasmic connections through which water can pass. The aquaporins are water channels, and these water protein channels just increase the rate of osmosis, but they do not affect the direction of osmosis. Of course, sugars and amino acids need carriers or transporters to be carried from one cell to another, whereas the ions require their specific ion channels. Let's see this interesting example about how the Turgor pressure affects water potential. Just like we saw before in our famous example of the U-shaped tube, we said that if you apply pressure from the outside, in addition to the atmospheric pressure, this would affect the flow of water by osmosis from the hypotonic solution to the hypertonic solution. This applies in vitro and also applies in plants, in animal cells. But here we're going to see how Turgor pressure will affect water pressure. In such a case, the Turgor pressure will be comparable to the pressure we apply in addition to the atmospheric pressure, or we applied in our previous experiments. Let's look first at the drawing to the left. We have here a container where they are just reminding you of the laws or the rules. You have a container, and that container is open. Is it important to say or to notice that that container is open? Yes, because in such a case, the atmospheric or the pressure applied to the surface of the fluid in the container would be atmospheric pressure, and therefore we consider it as if it weren't there. Now, if we come to, that's why psi p, or the pressure potential, would be considered zero, because by convention between the scientists, in order to standardize the way we calculate things, we have the psi pressure or pressure potential is zero because it's only atmospheric pressure, and the solute potential is also zero because this water is distilled water, and therefore it contains no dissolved salts. Now we are going to take a plant cell, and the plant cell will be immersed in distilled water for a while. Let's see the status of the plant cell immediately after being put in distilled water. Is it important to say immediately after? Yes, this means that we didn't give time to water to flow inwards or flow outwards. We didn't give time to water to flow inwards or outwards. So what's the original primary status of the plant cell? Of course, we have a Turgor pressure equal to, here, this psi p represents the hydrostatic pressure inside the cell. Why do we consider it 0? Because it is the reference value before entry of any water. So we consider it 0 because the plant is not turgid, it's not fully swollen. In order for it to be turgid, and in order to have turgor, when we say psi p equals 0, this means that there is no turgor pressure, not yet any turgor pressure. Why? Because the amount of fluid inside is not enough to press the cell membrane against the cell wall. So the turgor pressure is 0 and psi p is 0, whereas, of course, the solute potential is negative because there are particles of salt or sugar that are dissolved in the sap, and therefore it is -2. But after having been put in a hypotonic solution and after water entered from all sides to the inside of the plant cell, the sap vacuole became swollen. And when the sap vacuole becomes swollen, this means that the hydrostatic pressure inside the sap vacuole increases, and there will be pushing on the cell membrane, pushing from the cell membrane onto the cell wall. So psi p here represents again the Turgor pressure. Psi p here is the Turgor pressure, and you can see that its value has increased to +2. When its value increases to +2, it antagonizes the value of the solute potential so that water potential becomes 0. Water potential inside the sap becomes 0. If the water potential inside, of course, we're talking here about the water potential inside the sap vacuole. If the water potential inside the sap vacuole becomes 0, and the plant cell is surrounded by distilled water, which also has a water potential of 0, then they would be equal, and flow of water to the inside of the plant cell will diminish. So over time, there will be enough positive Turgor pressure. So whenever we say psi potential in an equation on plant cell, we mean Turgor pressure. It builds up to oppose the more negative solute potential of the cell. This we have explicitly explained on the previous slide. And eventually, the water potential of the cell becomes 0. So it becomes equal to I'm reading what's the text on the slide above the drawing. It will equal the water potential of the pure water. Both of them will be 0. At this time, water flow will stop. But why did it stop? It apparently stops, but there is a dynamic equilibrium. What do we mean by dynamic equilibrium? We mean if this is the sap of the plant, and if this is the outside distilled water, so this is inside the plant, and this is outside the plant. So at any time, there will be an amount of water that enters and that is equal to the amount of water that leaves. So whenever the status of equilibrium is reached in any context of osmosis, this means that apparently osmosis stops because the volumes of water on both sides, or the concentration of water actually on both sides becomes constant or stays constant. That's why we call it equilibrium. But it is dynamic, meaning that it's not static, meaning that there is inward flow and outward flow. But at any time, the inward flow is equal to the outward flow, and this is the status of equilibrium we have talked about. For example, here we have pure water that has psi p 0 because the container is open and exposed to atmospheric pressure only. Psi S 0 because there are no dissolved salts. But for the potato cell, the water potential is -3. Why? Psi P equals 0 because we are still exposed to atmospheric pressure only, but psi S equals -3 because there are salts dissolved in the sap, because there are salts dissolved in the sap. So the algebraic sum is -3. And then over time, what happens? Over time, pure water potential stays as it is 0, but the potato cell water potential becomes also 0. So we reach a state of dynamic equilibrium. And this is just because while the solute potential stayed as it is -3, there has been a significant increase in psi p, which is the Turgor potential, and that significant increase resulted from what? Resulted from the inward flow of water by osmosis. So the two examples, the one on this slide and the one on the previous slide, are exactly similar to each other. And now we talk about the aquaporins. As we said before, the aquaporins are water channels. So they are specific to water, and they are protein channels as usual, because water is polar, and therefore they should be hydrophilic from inside to allow the passage of water. So the presence of aquaporins would just increase the potential of water to pass through the cell membrane, since actually water molecules can pass easily because they are small molecules and they are polar molecules. The aquaporins are, polar. The aquaporins are considered pores in the membranes, but actually they are not pores, they are channels. And they are present in almost all biological cells. So you will find them in eukaryotic animal and plant cells, as well as in prokaryotic cells. The aim of aquaporins, whenever they are present, is to facilitate passive diffusion, of course, of water, and passive diffusion of water, you can just say osmosis. So they facilitate the passage of, or the osmosis of massive, huge amounts of water across the cell membrane. But they do not affect the water potential gradient, meaning that if you have a certain potential gradient or difference between, as we explained before, a hypotonic solution and a hypertonic solution, there is a certain difference that allows a certain rate of osmosis or a certain rate of flow of water between the two solutions. We said before that if we apply pressure here, this is one-way hydrostatic, mechanical pressure, this will increase the rate of osmosis. If we increase the osmolarity of the hypertonic solution, this will increase the rate of osmosis. These are two means of increasing the rate of osmosis we have talked about before, but we can also increase the rate of osmosis by having aquaporins in the cell membrane. So they will not change the gradient, but they will change the rate of osmosis. Similarly, they will not affect the direction of water flow, meaning that water will always flow from a hypotonic solution to a hypertonic solution. But they will only affect the rate at which water diffuses down the normal concentration gradient. And actually, instead of saying affect, we can say augment or increase, because they always increase the rate when they are open. We can consider them as gated channels. Why do we consider them as gated channels? Because just like in ion channels, they will open and close according to in response to certain stimuli. And the stimulus here is a Turgor pressure. For example, if the turgidity inside the plant cell is high, then the aquaporins will a bit close to minimize the entry of water by osmosis. So gated channels, as we've said before, are protein channels that open and close under certain conditions. If there is one of the major triggers for the aquaporins to open or close is the sudden changes in cell tonicity. If there is a sudden decrease in the cell tonicity, this will do what? Let's write it down in order to avoid confusion. So we are talking now about the cell tonicity, meaning the tonicity inside the sap vacuole. If the tonicity increases in the sap vacuole, this means that we need to attract more water, so the aquaporins will open, and vice versa. And now we're going to talk about active transport. Active transport is exactly the opposite of passive transport. We have studied active transport before in the middle school and in the high school. Active transport is transport that requires energy. So the main determinant is the need for energy. And therefore, it will occur against gradient, against chemical gradient, against electrical gradient. For example, against chemical gradient, meaning that it will occur from lower concentration towards higher concentration. Against electrical gradient, we usually find electrons moving from an area where they are more abundant, an area that is more negative, to an area where they are less abundant. The area where they are more abundant is the negative pole, and the area where they are less abundant is the positive pole. So electricity normally flows like this. But if it happens that electrons move against electrical gradient, meaning that, for example, you have positive charges or an overall positive status outside and an overall negative status outside, and you find that electrons or negative charges still move inward, so this is against electrical gradient. We have two major proteins that act as active transporters, the pumps and the carriers. Concerning the carriers, we'll be studying them in full detail in mitochondria and chloroplasts, because the carriers, the most famous carriers are the electron carriers that are present on the inner membrane of the mitochondrion, or that are present on the thylakoid membranes of the chloroplasts in the grana. And for example, the best example of an electrical carrier is plastoquinone. Plastoquinone is considered a mobile electron carrier because it can move along the plasma membrane. But you will find that many of the electron carriers, say for example that this is the first protein electron carrier on the inner membrane of the mitochondrion, and this is the second one. So these electron carriers transfer electrons from carrier number 1 to carrier number 2, and then carrier number 2, as we will see later on in full detail, will carry the electrons to number 3. But meanwhile, you will find out that those carriers on the inner membrane of the mitochondrion, or the inner mitochondrial membrane, for example, are not only mere carriers, but they are also hydrogen pumpers. So whenever you are to describe, or you're asked to describe the electron carriers on the inner mitochondrial membrane, please don't forget that they carry out another important function, very important function, which is that they pump hydrogen ions against concentration gradient, and this is very important for the process of respiration in mitochondria or photosynthesis in chloroplast to proceed. And if you stop the function of hydrogen pumping, the whole process will proceed. And then we have another famous active transporter, which is the sodium-potassium pump, and we've talked about it before. And we said that it is an integral membrane-bound protein, meaning that it spans out the whole thickness of the membrane, that it is present in higher animal cells, that the sodium-potassium pump is always related with another protein, which is an enzyme called ATPase enzyme that hydrolyzes ATP into ADP plus inorganic phosphate plus energy, and the energy released will be used in active transport. And the sodium-potassium pump is very specific because it pumps, and when we use the verb pump, we mean that it is an active transport mechanism. So we cannot use the verb pump unless we have an active transport process. It pumps both sodium and potassium in against concentration gradient, and that's why it is a pump. They are pumped against concentration gradient, meaning that sodium is pumped from from lower concentration to higher concentration. Sodium is pumped from the inside of the membrane or from the inside of the cell where it has a lower concentration to the outside of the cell where it is higher in concentration, and potassium in the opposite direction. So it is a co-transporter because it transports two ions at the same time, and it is an anti-transporter, as we said before, because the two ions are transported in opposite directions. And in addition to this, the number of ions pumped is not the same. We have 3 sodium ions that are pumped outwards for 2 potassium ions that are pumped inwards. One of the major significant functions or the importance of the sodium-potassium pump is that it keeps sodium ions mainly extracellular, and it keeps potassium ions mainly inside the cell. And this is one function, and this is important for proper functioning of the cell. And we said this before, but the second function is to create a cell membrane potential to make the outside positive in relation to the inside. And this is the state of polarization. Polarization meaning that even in an unstimulated regular cell, the outside of the membrane is always positive in relation to the inside. And this is what this difference in potential is what we call the cell membrane potential. I'm going fast because we have said this before, and you can review the previous videos in order to understand more about this. And then we have a third type of, so we discussed the electron carriers as one example of active transport, the sodium-potassium pump. And number 3, we have the glucose-sodium co-transporters. Notice that glucose carriers are concerned with a special type of passive transport called facilitated diffusion. These are simple glucose carriers, but there are other glucose carriers that combine the passage of glucose with the passage of sodium. These are active ones, and they are called glucose-sodium co-transporters, and we will be discussing their action later on. So glucose can be transported inside the cell either by facilitated diffusion, by carriers, normal carriers, simple carriers. So if glucose is transported via a simple carrier that transports glucose only, so this is a simple carrier that transports glucose only, and that does not require the use of energy, and that it is down concentration gradient, and therefore it is a process called facilitated diffusion because it is facilitated by a carrier. But there is another transport carrier or protein that is called glucose-sodium co-transporter. This glucose-sodium co-transporter does not transmit glucose only, but transmits glucose, and at the same time, it transports sodium. And it will require energy in part of its process, and therefore it is not facilitated diffusion, but active transport. So if you're ever asked about the ways of transport of glucose, don't forget that it might be transported passively by facilitated diffusion or actively by active transport. We have also seen the contractile vacuoles at the beginning of our study of the histology of the cell and or the structure, sorry, of the cell. And we saw that pumping out of water, contractile vacuoles need active transport to pump out water. We have also seen that certain secretions are in vesicles that are punched out of the cell membrane when we talked about the negative equilibrium of the cell membrane, or negative membrane equilibrium, meaning that we are losing part of the membrane to form a vesicle, and that vesicle separates from the cell afterwards and carries a hormone or a secretion. This process is called exocytosis. It is carried out when Golgi apparatus approaches the cell membrane and releases the vesicle, and then the vesicle fuses with the cell membrane and then punches out of the cell membrane. We have discussed all this before. This is an active transport because the mere movement of the vesicle inside the cytosol is an active process. Similarly, opposite to exocytosis, we might have endocytosis, in which case the cell membrane invaginates to accommodate the molecule that wants to enter the cell, and then a vesicle is formed, and then this vesicle would move inwards. So both exocytosis and endocytosis are active processes, and we also have the bulk flow. I don't know whether we are going to study it in detail. Yes, so we will be studying it in detail later on. The bulk flow is a type of active transport. We will stop here and then we will continue in our last video of, hopefully the last video of about the cell. Thank you.